This is week 1 overview of genetic tests. The learning objectives are to distinguish between types of genetic tests, including chromosomal, molecular, and biochemical, to compare and contrast diagnostic and screening tests, giving examples of both, to give examples of numerical and structural chromosome abnormalities, to recognize major chromosomal abnormalities and their association with major syndromes, malignancies, and spontaneous abortion, to demonstrate standard knowledge of cytogenetic nomenclature, to distinguish different banding techniques and their specific applications, and to use the International System for Human Cytogenetic Nomenclature, ISCN, to decipher and correlate karyotypes with clinical features and natural history, and to read and write karyotypes. In looking at genetic testing, gene-based technologies have had a major impact on the diagnosis of disease and are revolutionizing medical treatments and the development of specific and effective pharmaceuticals. Researchers are identifying genes involved in both single gene diseases and complex genetic traits, such as in breast and colon cancer. Using DNA-based tests, scientists can directly examine a patient's DNA for mutations associated with disease. Gene testing was one of the first successful applications of recombinant DNA technology, and currently more than 900 gene tests are in use. Most tests are for single gene disorders, but some, like the tests for breast and colon cancer, are for complex disorders. Genetic tests are administered at all stages of human existence and for a variety of reasons. Identifying mutations can help in diagnosis and choosing treatments, such as when a person only has some symptoms that are part of a recognized syndrome. Results of a genetic test are unlike the results of a cholesterol check or an X-ray because they can have effects beyond the individual to family members who share genotypes that affect health. As the pace of exome and genome sequencing accelerates and such testing is more widely available, it may become more economical to obtain all the information and parse it for results relevant to an individual than to do single gene tests. Cost is based on several factors, including the effort of sequencing and interpreting information. Obtaining a so-called raw sequence of a genome may cost under $1,000. Yet, an analysis of every known variant of the BRCA1 and BRCA2 genes costs more than $3,400, for example. If we look at the next charts, we're going to be able to see some of the tests that we look at. And if we look at them as far as a developmental time frame, we can see that these tests are at different periods of individuals' lives. This first slide shows tests that are given during the prenatal period, the newborn period, as well as in children. The examples on this slide are for tests for adults, as well as those tests that are given post-humerus. In looking at preconception and prenatal testing, some prenatal genetic tests have been in use for decades. Others are new or in development. Some examples are chorionic villus sampling, CVS, maternal serum markers, amniocentesis, and non-invasive prenatal diagnosis. It's been possible for many years also to collect sperm and separate them into fractions that are enriched for X-bearing or Y-bearing cells to attempt to conceive a girl or boy. We will be coming and discussing more later on about the prenatal genetic tests. But for the moment, we're going to move on and just give more of a broad idea of the types of tests that are available currently. The goal of newborn screening is to identify infants who are at high risk of developing certain inherited diseases that are so-called actionable because they can be treated or they can be provided with services to improve the quality of life. Testing a few drops of a newborn's blood for metabolites, which are biochemicals that indicate an inborn error of metabolism, has been routine for decades. For most conditions, an analytical chemistry technique called mass spectrometry detects abnormal metabolites, but DNA testing has been added. The number of conditions screened for in newborns has steadily grown from the first one tested for PKU, which was phenylketonuria, to more than over 60 conditions today.
The field of newborn screening began in 1961 with the Guthrie test for fetal ketonuria, or PKU. It detects phenylalanine, which builds up in infected individuals. It's an autosomal recessive disorder. In 1963, a specialized diet became available, and as we talk about this diet in a few of your other classes, you'll see that it's a difficult one to follow. It's easier when the children are little and you can watch everything that they're eating, but as they get older, it's much more difficult. And the diet does prevent mental retardation. So prior to 1963, we had no way to be able to feed these children until this diet was introduced.
After the diet's success, genetic tests expanded, and some states today perform DNA tests on newborns as well as biochemical tests. This next slide on newborn screening shows that in addition to taking a newborn's baby's footprint, a few drops of blood are sampled with a heel prick for metabolic and genetic testing. In looking at the genetic testing of children, we see that the tests on children range from single gene tests that are done because symptoms match those of a known disease, to chromosome tests, to exome and genome sequencing. A chromosomal microarray analysis detects tiny deletions and duplications that are associated with autism, developmental delay, intellectual disability, behavioral problems, and other phenotypes. If a parent has the deletion or duplication and is healthy, then it's considered harmless. Finding a deletion or duplication that a parent does not also have might be comforting because it may explain the child's problems, although it wouldn't necessarily change the treatment. In looking at genetic testing of adults, like children, adults take single gene tests as part of a diagnostic workup based on symptoms or other test results. They may also take genetic tests to detect increased risk of developing a particular cancer, such as a BRCA test. Many adults take direct-to-consumer DNA-based ancestry tests, and DNA testing is commonly done in forensics. In the military, genetic testing is used to identify remains and to detect a disease that might put a soldier at risk. Many adults begin to think about genetic testing when they are considering having children and wonder what traits and illnesses they might pass on. In the past, carrier tests in adults focused on specific population groups in which a disease is more common. In the 1970s, carrier testing for sickle cell disease targeted African Americans, while testing for Tay-Sachs disease targeted Ashkenazi Jewish population. However, faster DNA sequencing has lowered the cost of carrier tests at the same time that populations have become much more ethnically mixed. As a result, gynecologists now offer tests that identify carriers of more than 1,000 recessive disorders to patients considering parenthood, performed on one blood or saliva sample per person. If a woman is a carrier of a specific disease, then her partner can be tested too. If we look at the types of genetic tests, we see that the genetic tests can be part of a research protocol, or they can be regulated by government agencies. Healthcare professionals can recommend genetic tests to identify people at increased risk of developing an illness or to help diagnose a disease. Or some of the types of DNA-based tests have been offered to consumers by for-profit companies. Companies have marketed direct-to-consumer testing for traits, susceptibilities, and genetic diseases. In the United States, the Clinical Laboratory Improvement Amendments, or CLIA, control genetic testing of body materials for the prevention, diagnosis, or monitoring response to treatment of a disease or health impairment. The CLIA regulations, instituted in 1988, added specialty areas in 1992 to cover very complex tests, such as those involving immunology or toxicology. These did not include genetic testing, which at that time was limited. Since then, the genetics community has asked that the genetic test be included as a specialty area. State regulations can override CLIA, but only if they are equally or more stringent. Therefore, the regulation of clinical genetic tests can be unclear and still remains unclear in many areas. Availability of exome and genome sequencing will complicate the regulation of testing because of the increased amount and depth of information that sequencing provides. This slide lists genes that have variants that could impact such characteristics as muscle response to exercise, metabolism, tendency to gain weight on specific diets, endurance, strength, and susceptibility to specific injuries. All of these are being used to see if someone would have an inborn athletic ability. So several dozen companies worldwide offer these direct-to-consumer genetic tests for athletic ability. The customers mail in saliva or cheek brush scrapings that yield DNA and receive lists of variants of specific genes a few weeks later. And if we look at two particular genes that have gotten the most attention in predicting athletic ability, we have ACE, the angiotensin 1 converting enzyme gene. This is a gene that has shown in several studies that it is more common in individuals who do endurance activities, such as people who do triathlons, as well as among elite mountaineers and in populations who live at very high altitudes. We also can see some elite swimmers have some of the variants of this disease, which increase their power and strength with the training. The second commonly discussed athletic gene is ACTN3, alpha-actinin 3, which encodes a protein that binds actin, which, as you all know, is part of muscle contraction. We see that there are some genotypes that are more common among elite sprinters and another group which is found among endurance athletes. Some parents are testing their children for gene variants associated with athletic ability and using the results to decide whether the child should pursue a sport that entails sprinting or endurance. While some geneticists warn that DNA-based tests for athletic ability are too simplistic and illustrate genetic determinism, the company websites do have caveats and limitations and stress that gene action combines with environmental factors. Today we also have nutrigenetics testing. There's websites that offer the direct-to-consumer tests along with general questionnaires about diet, exercise, and lifestyle habits. The companies return supposedly personalized profiles with dietary suggestions, often with an offer of a costly package of exactly the supplements that an individual purportedly needs to prevent realization of his or her genetic fate. After the media spread the word of these services a few years ago, the U.S. Government Accountability Office tested the tests. An investigator took two DNA samples, one from a 9-month-old girl and the other from a 48-year-old man, and created 14 lifestyle dietary profiles for these fictitious consumers, 12 for the female, 2 for the male. The samples were sent to the 4 nutrigenetics companies, none of which asked for a health history. So we can see that these can be quite dangerous. Individuals react differently to the same dose of the same drug, even accounting for weight, gender, and age, because we differ in the rates at which our bodies respond to and metabolize drugs. Genetic tests can identify these differences. A pharmacogenetic test detects a variant of a single gene that affects drug metabolism, and a pharmacogenomic test detects variants of multiple genes or gene expression patterns that affect drug metabolism. Pharmaceutical and biotechnology companies use these tests in developing drugs, and physicians are increasingly using these tests in prescribing drugs. Genetic testing can guide the drug selection. And this offers several advantages: identifying patients likely to suffer an adverse reaction to a drug, selecting the drug most likely to be effective, monitoring response to drug treatment, and predicting the course of the illness. We will be looking at the pharmacogenetics in much more detail as we go through this part of the course.
Please watch the 2 videos on genetic testing, read the NIH fact sheet on genetic testing, listen to the presentation on the role of the genetic counselor in the laboratory today, and read the article on the role of the genetic counselor in the laboratory, and then go to the discussion board.